We are starting with the scientific evening, this time dedicated to one truly magical molecule which goes under the name DNA. We all know it. It's truly magical because it makes matter to be aware of itself and the universe. It makes matter to replicate and build these beautiful biological systems. So this is why it is magical. So when in 1953, Watson and Crick, in an old English pub, which is here on the slide, constructed a double helix of the DNA structure and show it to the world, they studied with fairly simple X-ray diffraction technique. That was not supported by a strong computational power and huge databases that we have it today, but still they managed to resolve these structures. However, the pioneers of today, they are further investigating the beauty and the secrets of this molecule by new, advanced, sophisticated techniques. And our guest today, Professor Plavec, he's using nuclear magnetic resonance to reveal hidden secrets of DNA. So please allow me, before I invite Professor Plavec to take the stage, to introduce him with a couple of sentences. Professor Janez Plavec is the head of the Slovenian NMR Center at the National Institute of Chemistry in Ljubljana and professor of structural biology at the University of Ljubljana. He obtained his diploma in 1987 at the University of Ljubljana Faculty of Chemistry and Chemical Technology and received his MSc degree in 1990 at the same faculty. His PhD degree was conferred by Uppsala University, Sweden in 1995. He was a Fulbright Fellow at Georgia Institute of Technology, Atlanta, USA in 2002. Research interests of Professor Plavec include studies of structure and dynamics of biomacromolecular systems with NMR, structural studies of nucleotides and nucleic acids, protein structures, interaction of small molecules and metal ions with DNA and RNA, and so on. His recent projects have been focused on NMR spectroscopic studies of small organic molecules and biomolecular systems, structural characterization of prion proteins in relation to prion diseases, structure and dynamics of G-rich DNA segments, cation binding and interactions of small molecules with nucleic acids. So Professor Plavec has co-authored over 200 original scientific papers, over 100 invited lectures at international conferences. He was a mentor of 14 graduate students, and in 2017 he was awarded the Choice Award for excellent achievements in structural studies of nucleic acids with nuclear magnetic resonance, and in 2018 he obtained the prize for excellence by Indian Society of Chemistry and for chemists and biologists. So Professor Plavec, we are delighted to have you here, so we are waiting for your talk now. Thank you.
Thank you very much. It's a very nice introduction. It's a great pleasure and honor. I'd like to thank a particular rector of university, Professor Zavrtanik, because indeed it is a great pleasure always to come to this part and also giving me this honor to present. I must give you credit, it does not happen. In fact, it has not happened that somebody would know where this is. And but this is, this is a rough pub, yeah? But I usually start like this because male audience would like to figure out what drinks are served. Am I too loud a little bit? Is the echo too bad? Your control. But ladies are usually subtle, right? And they look at details that men would not find out. And as it was said, in this very place, two important men, Watson and Crick, announced that they have uncovered the secret of life, meaning double helix and so on. I'll come to that. Let's go back to the bar tops, and you will be surprised. Where you have a pub, that they would, bar top would be a double helix with an eagle. it so happens this pub is called Eagle's pub and has been around for centuries, but it's probably most famous nowadays for this Watson and Crick story. And you see double helix on the bottom here. And as it was said, Watson and Crick made this place very famous. Introduction was made, so I'll not spare any time. You heard a lot about me. I gave this magic Čarobnost in Slovene. I thought, at least for me, it has like a little mist into it. I'll try not to bore you. I give a little bit of general introduction, what we are talking about. Then I introduce something we call G-kvadrupleksis, and I touch, as it would be appropriate, on 3 subjects. I give you little hints what we are doing and where we have contributed to scientific field recently. But of course, I'm free to discuss and open anything. I think it's appropriate to start with really where I'm coming from. Slovenian NMR Center, it's state. It's a research facility of this country. And it started years ago, when the major thing happened before my time, so to say. In 1992, Ministry of Science of this country decided to start an NMR center. And that has been it has been a very successful story. Even in the day, 94, 95, 5 academic institutions, 5 companies joined forces. Nowadays we would say PPP, right? But this, I'm very glad that this consortium still exists, to satisfaction of academia and industry, and I'll come back to this. A big step forward was made, and for locals we would know, where centers of excellence were established, because that was actually the last time, unfortunately 5 years or so ago, where this country finally, or at least gave some money into research infrastructure. We are suffering from. Sorry for, I don't want to sound critical, but just state the facts. And then, more recently, now it's a little bit more than 3 years, when we became part of something that's called CERIC, Central European Research Infrastructure Consortium. That's worth mentioning, because this is where we a little bit collaborate with University of Nova Gorica, with a few people here. Idea, I like it, and allow me a word or two for young people. There is no tradition of investment in large infrastructure. And typically, replacement value of spectrometers we have, it's like 6-7 million euro. So when will you collect in our country? So clever heads, as you would say, came together and said, why don't Italy, Slovenia, Hungary, Poland, Austria, Romania, and so on, Serbia, Croatia, join forces and somehow agree on what will be the major infrastructure they will develop for the consortium as a whole. Story does not end here. The consortium runs now, actually right now, call 8 is opened every half year, and we run under open access model. So whoever, from wherever in the world, you apply for time of these facilities and you will be granted, if successful, on peer review basis. It so happens that in NMR lab we go with this number of megahertz, and they go in hundreds. 800 megahertz is our biggest. It's a little bit outdated, unfortunately. Then we have a 600, and one of the 600s, which is vintage 94. We'll test some wine later. So I think seniors, they know what it means to run 20 plus years. And they ask you, how modern is your equipment? So sometimes it's a little bit sour. Allow me, I see young faces. This is not a lecture as you would usually see or hear. I'm willing to come some other time, but I give you a little bit hints what nuclear magnetic resonance can give, and I'm very glad to meet a new friend here. I guess you all know this molecule, ethanol. I know you run a program, and when you go after the Second World War, this is some years ago, right? They uncovered signal. And it's very interesting for younger. Physicists observe phenomenon, and they did not believe that when chemists write structure like that it actually makes sense. So you can read papers from 50s when they say, when we see 3 signals in our oscilloscope at the time, then the physicists said, well, what chemists are doing actually makes sense, which I find it quite nice. So you can relate a given signal to a given chemical subunit. This is ethanol, right? And they go with integral. But the real point I want to make, this was very exciting. Somehow the name came chemical shift. Probably nowadays we would call it differently. If you will even Google nuclear magnetic resonance, you'll probably see, oh, this is unsensitive method. And this is one message I want to convey. We can determine structure of a molecule if you have 2-3 microgram of sample. I mean, and this, I think you'll understand the message. Signal to noise sensitivity has improved greatly, and that has really and helps us to study small molecules, as was said. This is important for many industries, even local industries of our country, namely drug, pharmaceutical industry. Most importantly, for drug industry, you can study this in the solid state. You can take a pill, crash it, and study. It's important for material research, and I guess there will be people here, and I'll come a little bit to that. Today I will focus on sort of the other side, biomolecular structure. We can determine structure of biomolecules, such as protein here, and where NMR is unique in studying what we call dynamic phenomena. Let's assume that a given molecule has a little bit structural arrangement, and chemists will understand equilibrium. And now it's not that you observe a little bit and you have to deconvolute. You can observe signal change on a per atom basis, sometimes residue basis, so you know which details of structure have changed. And this proves to be extremely valuable. I could talk, of course, for hours why that would be valuable. And another aspect where NMR is unique, we can actually look at dynamics of the system. How are certain parts of molecule moving? Are they millisecond? Are they microsecond? Are they nanosecond movement? You might think this is curiosity of this crazy guy speaking to us, but this has been proven that enzyme reactivity, it's related to dynamic of the active site. So we have method which will not in general, how does this molecule behave? But on a given part, we'll see it more mobile, the other part maybe a little bit more constrained, less well-defined, we could say. So this is what technology allows. I showed you an NMR spectrum of an ethanol, the simplest, more or less the simplest molecules. What we are doing in our center, we do high-dimensional spectra, which enables you then to go into this sort of structure that were actually meant in introduction. Nucleic acids, small molecule structure, and proteins, protein complexes, and so on. So NMR Center, I'll not bore you with detail, but just a little facts, because I think they're relevant. Annually, we would have 80 programs, including international programs. Nowadays, I mentioned CERIC, how people come in touch. Problems that we typically tackle are protein structure, nucleic acids, protein folding. I mentioned pharmaceutical industry. You try to understand how does the active ingredient of drug matters. Polymorphism. You know, for generic pharma, this is crucial. Do I have this form or the other form in crystalline? Biosimilars. You know, biogeneric, it's getting a big market, and NMR can help. Metabolites and complex. This is work of neighboring group, molecular sieves, storage, energy storage, you know, battery material. Our institute has very strong research group, and they use polymer departments. And coming back to wine, I admit to say I used to call it fun NMR. Actually, it's not my word. Other people use it, fun NMR, because 15 years ago, people would walk to the lab and take a little orange or milk they'd had in the morning, Coca-Cola. They just run an NMR. Nowadays, these are major activities because authenticity. We actually do this regulated by EU legislature. I put dots here because honey, olive oil, name it. To prove authenticity, I think we are all concerned what food we drink and so on. I said at the very beginning, I'm very proud to be here because I think this is the essence of being a researcher, being some sort of teacher. And another aspect is opening up. And I think what Professor Zavrtanik and his crew, sorry I named him, but opening up, we are not afraid to benchmark. So annually, typically, we'll have at least one event where we invite our international colleagues. So this year we are meeting in Bernardin. I hope they restore it by then. It will be October, which means you get your experts, they tell you you're doing right, and of course they also enjoy our beautiful country. I come to a subject. So DNA is in fact magic. So, I mean, it inspires. And I was introduced by how this structure was made, how it was arguably perhaps one of the biggest discoveries ever, because there were, of course, discoverers got a Nobel Prize some 10 years after discovery, but biotechnology, molecular biology, really, really different fields of science that, of course, were not available. It's interesting, so it's also very inspiring for artists. And probably you know that usually we say right-handed helix. And ladies wouldn't know, but men know. Right hand screw go that way, not left hand. If you buy yourself a little thing for your ears, it's usually left-handed. For some reason, artists like to make left-handed helix. And not that it's not relevant, I'll come back to that. So we all learn very early on in our education how these two strands, two molecules, are held together by a force that we call hydrogen bonds. So guanine would pair with cytosine with 3 hydrogen bonds, A would bind with T with 2 hydrogen bonds. And this is actually how things assemble, how they associate, how you become a successor, and so on. One thing that I want to point out, and this may be a little bit more real structure of it, one force is hydrogen bonding, and you can use this model to explain a lot. Another that's not so commonly perceived is stacking. You have these base pairs and they stack, usually 3.4 angstrom. And it's amazing. You change hydration environment, we can say, I'm coming back to this ethanol story. If you put molecule in ethanol, you dehydrate it. So maybe hydrogen bond recognition will be weaker. So another force of stacking will change. You change cation concentration, and then you get this balance of forces. So, however, I argued this is one of the biggest discoveries of this universe, of human race. It's very interesting. It's like a huge tree, and nothing grows under. But in fact, when you go back to 50s, you see that several people reported different type of structures that utilize different way of assessment. Triplex, quadruplex, I'm not going to, more and more. But the biggest discovery, double helix, was so huge that these things were somehow forgotten. And that's what I want to uncover. That's this magic I'm talking about. How there were these structures that were somehow there, but were not felt important for a moment. So when you look at any nucleic acids, the basic unit, you can consider sugar unit, phosphate, that's a backbone, and then you have 4 residues, 4 heterocycles. And I'm a chemist by education, and chemist understands that if you consider a given residue, how it will interact with neighbor. It will be through hydrogen bonding. And then anybody that had the class of chemistry will understand you have to consider hydrogen bonding ability and hydrogen acceptor ability. And this site of guanine is being utilized in GC base pair. So it's important for replication and so on. But you see all this other ability, and this has been proven by scientific papers. You can form ribbons, like 2-dimensional material. And it's been utilized, actually, this field, nanotechnology field, is quite big. If you consider these 3 rings, you flip around the 3rd one, and you add a 4th one, you can make a circle. Circular arrangement that's known in the literature as a G-quartet. Here you have a little bit more realistic views that come from crystal structures. One thing to mention here is, you see this red, these are oxygen atoms of guanine residue. They're electronegative, they repel. So actually you need cation to compensate. Mind you, and this is crystal structure, so it has been proven with high resolution that it's really sitting there. Mind you, sugar phosphate backbone, it's negatively charged. So you anyhow need cations for somehow a neutral condition. But in this particular, when you have guanine, you need additional cation to make structure, to make these quartets arrangements. And you wouldn't believe, first reports of these things were made in 1910. Watson Crick, 53. In 1962, there is a paper that there is something else going on. So can we now, and that was guanosine. just for a little bit, in antiviral field, lots of guanosine analogs are antivirally active. But acyclovir, if you have herpes, you would know it, or some other virus. So now let's consider if we make an oligonucleotide, which has several Gs, will that form G quartets too? And in fact, it does. Interestingly so, if you have this large aromatic surface, you get cation, they stack very well. Large aromatic surface stability. Now it's interesting to consider, we had a little bit resolution, so because you have 4 guanine tracks, you have to start thinking about, do all these go in the same direction? Because nucleotide has a 5' end and has a 3' end. So there is certain directionality to it. So if you have 4 G tracts, they can all run in parallel direction, or they can run in anti-parallel. And now you can already quickly imagine, if I have 2 plus 2, there are already a few ways how you can make this arrangement. And there is also in the literature, and actually quite important, 3 plus 1 arrangement. That's the terminology of the field. And now you can imagine if you have an oligonucleotide, there are at least 5 possibilities how these G3 elements will orient with respect to each other, forming a G quartets, and this symbolizes the direction of the strands. Then we have these wiggly parts. These are in this field called loops. They're meant to connect two G-rich tracts. And how would you do that? And you can envisage, you can have H-type loop, diagonal loop, double chain reversal loop. So I showed 5 ways earlier. Combine it with at least 3 basic canonical type of loops, and you can imagine a lot of possible ways to fold this. Actually, this is polymorphism, the way we know it from crystallography, and that makes this field quite challenging. And I'll show you some examples later. You can take a given oligonucleotide, make a small modification, and structure will change. I don't know how well you can see details, but it's even not so important here. Some years ago, Maya was my student, and we were very lucky. We worked on the sequence that actually comes from human papilloma virus, and we were lucky enough that Maya uncovered a structure that has all 3 basic type of loops in a single structure. That was the first at the time. It has this propeller, double chain reversal loop, diagonal, connecting the diagonal of a quartet, or edge. What I want to argue here is, one is we say topology. So how do you connect? But there is more. NMR and high resolution structure tells us more. It gives atomic position in this model. And why would that matter? Because a given residue can co-stack. It means it contributes to stabilization of overall structure, or it can stick out. It can be exposed to solvent, representing a potential receptor, potential site where it will interact with other molecules. And that's what we want to argue. So we have methodology, nuclear magnetic resonance, that can, with the help of calculations, actually, this was mentioned early on, we can get these models that might be useful. I will not come back to this, but of course, idea is if you have viruses have ability to form these very unique structures, we try or want to exploit them for development of vaccines, for example, or drug that will stop deadly disease. So I mentioned some dates here and there. And this was considered till 1980s curiosity of chemists. And then things started to come out with the genome of many species, including human. And it is uncovered that telomeric ends, this is the very ends of chromosomes, are guanine-rich. Idea being that guanine-rich region, when you replicate, when you divide cell, they do not replicate. They get shorter and shorter. And in fact, that represents molecular clock. But you know, aging, there are lots of ladies here, right? You want to stay young, right? Forever, hopefully. I'll come back to that perhaps, or later in discussion. So we have a way, as cells divide, part of the G-rich track, which is several kilobases, the variant is like 200 of those, get shorter and shorter. So it's like molecular clock. One is too short, certain processes, senescence, apoptosis start, and cell dies. And then when things became really interesting was around 1980s. They discovered telomerase. This is an enzyme, it's a reverse transcriptase that takes an RNA template to make DNA. So it literally glues back these G-rich repeats. And of course, bad thing is it's upregulated. So it means works very well in cancer cells. And you see, this was important discovery too. These are Nobel laureates that got their Nobel Prize for discovery. Interestingly, if you have G-quartets, and I'll show you a little bit how they look, you have a way to inhibit telomerase. And you can understand interests of pharma, develop small molecules that will recognize these sites or even stabilize them, just to make this cancer cell, their proliferation controlled. That's the beauty of it, right? And coming back to Yuke's story, so if we have such a deadly beast, and we want to stop it, but what if you can control it well enough that you can revive cell? And actually, it's quite interesting. Blackburn was a supervisor. She believed this story is more important. And Greider, they actually both started business. It's quite an interesting story. Greider believes, well, I might have a way to control this beast to my benefit. I want to rejuvenate. I want to renew cells. And it's quite exciting. Of course, whenever you play with this in the body, you're on the sharp edge. I will stop with this story, but I'll show you a little bit how last 30 or so years, we know about it recently, I mean, it's 5 years ago, researchers from Cambridge, just by chance, have uncovered with some antibody, they discovered that ends of chromosome are really forming G-kvadruplex. That's a big thing in the field. Our G-kvadruplex is a little model that we are using to explain certain facts, or do they really exist? And this is one of the studies that sort of proves that that's the fact. Then, a little bit more than 10 years ago, this is the major paper, 2007, people start looking at the genome, even of human and other species. They say, well, we now know for a given sequence to form a G-kvadruplex, I need 4 G-rich tracts. And I allow anything from 3 to 5, and then I allow up to 7 residues to connect them. And they searched the genome and surprisingly discovered almost 400,000 of hits. It's a huge number. Of course, first question after that is, are they just sporadically? If they were, so-called density would be 0.1 per kilobase. But of course, when you do this bioinformatics in a computer, you quickly see, well, these are actually concentrated at sites near transcription start site, so where the genes are. And these are regulatory regions. And you see, 0.1, 28. So it's like over 200 times more. So actually, the G-rich regions are concentrated, if I can use this word, in the regions that control expression of gene. And of course, these are again some important disease like oncogene. These are genes that might cause cancer, and I'll mention one later. Idea being, if you have normal structure, normal in the sense double helix, everything works, you get a protein. If you have somehow quartet formation, of course, all these proteins that recognize the start and start expression, they will see different assembly. And believe me, there's huge interest also here. How do we make this stable if we can stop expression of a given protein? And it works the other way too, right? If I want to make something, I want to resolve this. And there are experiments that prove that. So I gave you this number, 376,000. I think this is a paper that gets some merit because it comes from sequencing by Sir Shankar Subramanian. He sequenced a given sequence, oligonucleotide. He's really expert of this in the presence of sodium ions. There are normal reads when he has potassium ions or some molecule that they call pyridostatin. There is change in sequencing. And with this study, which is more somehow experimental, not just bioinformatic, you see the number is even higher. So over 700,000 regions that have ability to form G-kvadruplex. Ability. usually in the field we say potential for quadruplex formation. So I think I gave you enough sort of introduction that you believe me, there is something to it, right? Let's try to understand what's going on. And here it's another fundamental thing that actually, at least in my eyes, deserves some merit and further study. See, this is 4 repeats of human telomeric repeat. TTA, GGG, 4 times more or less. Only the initial T's are not there. So they will form the 4 G tracts quartet, quadruplex formation. This structure, it's a sort of basket. If you ever go to market, it does look like a basket. The background is, it was determined as the presence of sodium chloride by NMR. Crystallographers, Nature paper, you must know it's quite important, the same repeat in the presence of potassium ions and crystal structure. And you already see some different color, you see the shape, more disc shape, more basket shape, top view, and I said earlier, we want to utilize this for drug development. If you want to put any rationale in it, these are really, really like day and night. So we want to understand what's going on. Is it methodology or is it the real cation? And this will matter. In certain cells you have more sodium, or certain compartments of cell more sodium, in the other you have more potassium. You will probably say, well, the first question is easy. Why don't we just take this and dissolve it and see what's the structure in potassium? And you will remember my earlier statements how polymorphic the structures are. And actually, in the set of structures, I know now I'm jumping to like my view of the current state of knowledge here, we have 8 structures that have been determined by high resolution. So I mean, we know by location of an atom with given precision, that actually come from 4 repeats of human telomeric repeat, more or less the same sequence. Now we can discuss later how is this relevant, but I think you can see that they are really different. So I just come a little bit back to NMR, how useful it is. This is again human telomeric repeat, and we can just screen for certain parts of regions of spectrum, and we can get these ratios. Is it quadruplex? It is Watson-Crick, it's I-motif, and other 4 standard structures. What we typically want to do, this becomes very useful for biological sets, because I showed you earlier on, by integration, you can get a ratio. You can say, oh, under this condition, I have preference of quadruplex over, what's, for example, duplex. What we really want in my group, or what makes us happy in a way, we make these sharp lines. This is like a dream of NMR spectroscopy. Once you reach this sort of state of your sample, you know you're, which we could say literally, it's an evening, right? You're in the game. So you know from here on, I'll be able to do structures. I'll give you a little story that actually it's quite relevant, and you'll see why. So this is human telomeric repeat, and there was this student that wanted just to work. He got the diploma already, and he was accepted to Singapore to do PhD. And that was like February, and he would go there in October. He said, I want to work with you. So what do you do? And I knew in Singapore what they do, and I said, okay, let's start playing with this folding. That was very happy. This makes me happy when other people can reproduce our work, or we can reproduce somebody else's work. So what we did, Blush, took this sample, added a little bit of cations, like 10 millimolar potassium chloride, and follow over time. Of course, initially, minutes, hours, days. We understood that it takes days to form the structure. What was very nice here, in a way, we beautifully reproduced that, in fact, structure that has been published years earlier is formed, but we also saw this sort of pink and black lines that are there, and actually, they're not, I don't know if I made this point very strong, they're not 3-quartet formation, they're 2-quartet. And somehow we said this might be relevant, how these structures form. So how would we get 2-quartet? We decided to somehow remove this final G. So we make G3, 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 and this one is just G2. We made this molecule and gives sharp lines. So we were very happy. We started working on it. One point I want to make. Don't take me wrong, but perception would be somebody reach, or you finally persuade somebody that it's worth putting €1,000,000 and buy a new spectrometer, and then it's easy for that person. No, it's not like that. We are not just drinking coffee and recording spectra and having fun with our friends. We actually need to work a little bit, and I show you how this works. So, you see, this is a molecule, and this is part of its proton NMR spectrum. How would you go and assign? Assign, in our words, means we know exactly what this iminoproton, to which residue does it belong. And we do partial isotopic enrichment, which means that a given residue, position 3, guanine 3, has been partially replaced by guanine that's N15 labeled. So you know N15 is only like 0.4%. We enrich it, and we record spectra. And this is the beauty of NMR. If only position 3 is labeled, we can run an experiment that only protons that are bound to N15 give a signal. And then we do it so you have to do many syntheses. What turns out of this, and I actually explained this already. What turns out that when you have position 5, there is no signal. So somehow this residue is not involved in hydrogen bond. So for others, we have 2 signals. So major and minor. This does nothing special. And for some, we have only minor. It's sort of hard to see from this one, but I'll show you later. And we worked on this, and we determined the structure. It turns out it's a monomer. It also looks like a basket that I was talking, but it has 2 quartets. Top one, bottom one. This is directionality of hydrogen bonds, anti-clockwise, clockwise, and we have these base triples. These are not so common structural elements, but obviously here they contribute to stability. Then there was a minor set of signals that I mentioned. Then we played a little bit with it. We decrease pH, we cool it down, and we saw that we go from one structure to the other, and some new signal shows up. So of course we were very interested in what's going on. More or less did the same process. Here it's something that, even if you're not an NMR expert, I hope you grasp. I want you to grasp this. So this is black. This is this new species. I show you one structure, right? This is position G3, it's labeled G4 and so on. And these are parts of 2-dimensional spectral that are called HSQC. And down here it's proton chemical shift. Proton, along this direct dimension, it's carbon. So like H8C8 pair, aromatic pair of carbon and proton. And NMR is extremely sensitive to changes. So now I show you the spectra overlaid of the species I showed you. You see red and black, they are really different. This means, in our jargon, shielded environment, which you can just consider like electronic environment of that proton and carbon has been changed a lot. It's changed a lot. So these are really distinct structures. What was very interesting, this new KDH plus also has 2 quartets, anti-clockwise, clockwise direction. But that's what I said earlier for the other form. It's a little bit confusing, right? So what has changed? And it's actually quite amazing. In this one, G22 is hydrogen bond donor to G10. Here is the other way around. Is G10 hydrogen bonded to G20? Like you would reshuffle them. But you saw earlier it takes days to fold. And then you can switch like that, change a little bit pH and cool down. That's quite exciting. So, one warning, and this is maybe for non-experts, but it's more warning for experts. If you do CD spectroscopy of these 2, I hope you believe they're really distinctly different, beyond doubt. That's why I made those ones. CD spectra overlap. So if you use normal, low-resolution spectroscopy, you will miss it. Temperature stability, it's 10-degree difference, which is quite exciting. And this is the 2 structures. They're both basket type. So what is different? You go down here, out of the plane and continue. Here you go down, you stay in the plane and continue. So you would think, and I did too, they look like mirror image a little bit, but they do not. You cannot think that way. So we took formalism for our colleague that he has published to distinct the 2. One thing is interesting, 820 gets protonate, that's why we put H2, and we run equilibrium to this side. And that's how we did it, right? TD was predominant at neutral pH, we decrease pH, that one goes down, KDH plus the other foreign goes up. And obviously you want to know, can I reverse it? Yes, we can reverse it. For non-expert, allow me, pK of adenine, it's around 4.5, but here, apparent pKa, it's around 6. This comes to a region when it might be biologically relevant. Obviously, you'll ask yourself, well, is this repeatable? Yes, it's repeatable. We did it many times, over 10 times, and we can run this equilibrium up and down. Why do I think this is relevant? Cancer cells are known to be a little bit more acidic. And we have brought to the field new way to think of these structures. We need cation ions present, potassium ions present, but we have a little shift of pH that can drive equilibrium. And it's a new finding. And Angewandte Chemie for chemists, it's a respectable journal. Ooh, I'm sorry. I'll rush up a little bit. I'm sorry. I don't want to abuse your hospitality. I mentioned a little bit oncogenes. So if you Google CIMIC, you will see that's a beast. It's responsible for many cancer. Here we use it as a model. It forms a parallel-stranded quadruplex. Structure has been determined earlier by my colleague Danzo Yang from Purdue University, and this is the spectrum. So we have a nicely reproduced her data, which is great. What I want to tell you about is this story. So this is a molecule that's called by its author, the OTAM2. This is triangulinium with this morpholino. Spectrum is here, everything is nice. Why we got interested in this is because my friend now, Ramon Villar, has got in touch with me and he said, well, I found out when I have this ligand and I bind it to different secondary structures of nucleic acid, when it's bound to G-kvadruplexes, fluorescence is longer lived. And this is really exciting. I mentioned earlier about skeptics. Are quadruplex really forming in living cell? And now he has made a fluorescence probe to the life cycle of cell. At certain, this S and G, and if you know more about this, certain stages they will lead more, in certain life cycle less. And this is also a wonderful proof. So our question was, of course, how do these ligands, heterocyclics, really interact with DNA? So typically, as you would in biophysical experiment, you start titrating, free, and then you add. You get to a point and you want to discard everything, but luckily you don't, you continue. At 1:2 ratio, we get a nice complex that we have solved to high resolution. This is a section or a region of two-dimensional spectrum that we call nosi. And that actually has been quite instrumental and informative because we could figure out how interaction of this ligand to one side is higher than to the other, and at the end we get a complex. I rush up because I want to show you something that's more recent and more exciting. So obviously we have certain interests, and I show you how we want to also understand basic properties of DNA. So you see this sequence, G3A, GCGA, and everything I showed you, this goes into the canonical expectation of the field that this will probably form a G-kvadruplex. This particular gene comes from a region in the 14th chromosome that's responsive for autism. So neurodevelopment, something goes wrong. So you have this repetitive sequence. So my student Wojciech at the time, he took this part, it's VK are his initials, and he said, well, if these are repetitive, we need to consider two possibilities. And then we start to play with this. One thing was interesting because we could consider if we have G3, GCG, we could have structure like that. And that actually, I still challenge even colleagues in my field, if we would publish it, they would accept it, because it's quite obvious. But then I showed you this paper before, where I showed you 700,000 structures and so on. Once we had our structure solved, we went back and they see some suggestion made there might not be the whole truth. Further studies or different type of structures need to be considered. So we folded this oligonucleotide, very much as I explained to you, did this, what we call sequential assignment. That means we go through this structure, nothing too unusual. I'm running out of time, but I want to show you the most exciting points. So you see this is obviously oligonucleotide that with all expectation even by experts in the field, would form a G-quadruplex, except that it does not. And what does it really form? We have in the core of the structure GC-GC pairs that pair to each other and actually stack. And on top of that, we have GA-GA pairs that also stack. They're dimer, and most exciting, this G3 element that with every common knowledge would be expected to form the basic unit of quadruplex, one of the strands. It's actually in the loop. They're paired, they are stacked, so that you see how they're parallel, but they are not. They're on the outside. Up here also, G3 elements, base paired, and somehow contributing to stability. But clearly, there is no G-quartet. That's why I say these are not G-quadruplexes. They're another family of quadruplexes. Here we made a little thing. We said if this is repetitive, can we make it a monomer? And it's a beauty. You add A16, and you see blue and green. You make it a unimolecular fold, and it's actually the same, which means to an expert, this word, it's actually a robust fold. Let's come to this other, GCG. So it's just shift register, because this, how will proteins, how will the molecule know? This one is even more exciting. Here we have GA-GA pairs in between, in the center. On top of that, GC-GC pairs contributing to stability. And again, G3 elements, clearly not forming G-quartets, but in sort of very new GG pairs with crisscross. I think it's a good English name, where you go 90 degrees between, I don't know how well you can see up here, but it's a sort of novel arrangement, clearly well stacked, so stability is not a problem, but obviously this is not a quartet. We were able to make this dimer and a monomer, because in biological context you accept this repeats to form a single molecule. So we had these two families of structures. We call them AGCGA quadruplexes, and really it's just you change your point of view somehow, right? And then Voigt said, let's see if this is the only this plec HH3 where we see this, and he did genome search and found like 50 different regions around our genome that have been actually reported. But of course, structure, even my medical doctor, neurodevelopment, this was somehow expected because autism, bone and cartilage development, cancer, and this I find most exciting. So we don't need to think of this magical, coming back to my word, right? We don't need to think of these unusual, non-expected structures as something wrong. No, I think we'll see that they're actually involved in normal, healthy cellular process. Coming to conclusion. Play, use NMR. We're actually offering hand, so you're welcome to join. That's the most important conclusion. Another is, I give you a little glimpse about human telomeric repeat, two types of structures that we covered that are two quartet forming. Otherwise, telomeric repeats are three quartets, and we had this nice pH switch from one to the other. I show you the complex of this drug. This was also in Gevante. You might think, what's so special about it? You wouldn't believe. You need some luck. This is work of Anita, my PhD student. I said she has a golden hand, because usually these things precipitate. You can imagine aromatic surfaces, they stack very well, so and then you have quadruplex, which also stacks very well. You're bound to get a precipitate, and you need some luck, not just knowledge. And I showed you this novel family of structures that we name AGCGA quadruplex. I showed this, why is this so interesting, and why we actually came across, and maybe this is maybe a little stimulation or incentive for youngers. You see, we had used look at this molecule. This is G3C. That comes from adeno-associated virus. We have studied this back in 2010. And in that case, we saw how water molecule is localized between G-quartets and GC-GC pairs. And that is still very exciting finding, because of course water is also another molecule, not of life, but something that surrounds us, it's very important. Let me acknowledge people that work with me on this. Wojciech, Anita, I mentioned their work, and there are several other. We had excellent postdoc from China. This field is great. I like to acknowledge help and collaborations with people around the world. I'm actually very happy that in this field we managed to collaborate within Slovenia. For foreigner, this might seem funny, but it's actually quite honorable thing. I tried to see, oh, I'm sorry, Ricardo, I thought I was going to say, today I'm joined with Ricardo Rico, who is spending a few months of his PhD in our lab, and I'm somehow forgot to, don't tell Claudia, I did that, I'm sorry about that. So we're a wonderful family. Of course, these are people that we meet to and discuss and obviously exchange students. I'm very grateful to Slovenian Research Agency for support, and of course everybody else for any money we can get to do this beautiful research. And with that I finish, and thank you very much for your attention. Thank you. Sorry.
It's a short review of chemistry of aging, isn't it?
You can see this one, yeah.
Yeah, so before I give the word to the audience, maybe I take advantage of my role today and ask you some quite a popular science question, which is, if we would be able to prevent telomeres, shortening of the telomeres, would cell live indefinitely that long?
Yeah,
would they die at all?
That's an interesting hypothesis. I see life has many ways. I don't know if I'll explain. You see, even cancer, it will be indirect answer, but even cancer. I told you this telomerase being uncontrolled and it just works like crazy in cancer cells, but it is 85% of cancer. There is something called ALT, so intuitive, right? Alternative mechanism. So this 15% of cancers go some other way. And that's my saying, a chemist view, life finds different ways somehow, because relations, they are so intricate, we get to understand. That's why, in my belief, these studies are important. We get to uncover little pieces of very complex puzzle. And I like your thoughts at the beginning. We need experts from any field. We need computation. What is becoming so popular now is big data. How does a given protein or a given nucleotide we look at, how does it really come into play in interaction? One thing though, if I come back, I find very exciting. I mean, this is DNA, this is our genome, this is our heredity, this is who we are. And when you get older and you see a young person on the street and you knew his parents, you say, oh, you are this guy's son or daughter, right? That's amazing. But when you grow up and cell divide, you go around the world, you eat different food, but somehow, when you get sick, it's unbelievable how this heredity, meaning genetics, works. It's amazing, right? Somehow information is stored. I show you regions that control how it's expressed, but this is, I guess, beauty. Somehow, this coding, I hope you understood today, we are not just reading three-letter code. We're not just reading DNA as a book. There are complicated, intricate relations. How does this molecule forward? And that actually controls how cell works, how life goes on. And I guess this is a little bit longer answer. I'm sorry, I'll try to be shorter.
So thank you for this. But now let's see if we have some questions from the audience.
It's very difficult to grasp. But you mainly show the proton with the hydrogen result, nothing on carbon or nitrogen. Where do you use this?
I did make this point that we do that. I did make this point that we do heteronuclear multidimensional experiments, but I thought that would be pretty boring today. But obviously we take advantage of every possible methodology, or in NMR they are called experiments, different pulse sequences. We utilize them a lot. But I thought for today, I give the most exciting, like creaming on the top. I mean, Wojciech, you will understand, Wojciech worked on first structure for 3 years because we went against all dogma. You wouldn't believe, we made one molecule isotopically labeled, the other one not, just to prove we have symmetric dimer. And then you have this intermolecular NOE between protons connected to isotopically labeled and being edited and then filtered to protons that are not. So there's a big array of experiments that are there to support what I was presenting. But this was like an NMR talk. I guess you and I would be excited. But
so, I didn't notice, so you plan to use some labeling to,
yes.
to place different...
Here, labeling DNA completely, it's too expensive. So we do partial isotopic labeling, but this is sufficient to somehow edit the spectra and get unequivocally assignment. I hope when some of the structure I show you, they're really novel. There's no precedence. So we want to be absolutely sure, by chemical way, we know where the label is and we see the signal. It's not like, no catch-22 here, right? If I assume, then this is. No. It's like ab initio. Really use an isotopic labeling, and then several experiments are being done. I said they're documented, they're reported, but...
In my times, we used deuterium. We developed a method through...
This is excellent point. This is excellent point. I didn't dwell on this. This is quite interesting, actually. You see, these structures by themselves, they're very dehydrated in the core. Here they look very open, but in fact they're not. So what we do, and they're connected quartet, or these new structures, they are connected through hydrogen bonds. So we do the trick. We lyophilize them, we remove all H2O, and we put them in deuterated water. And you know, exchangeable protons will exchange. And it's really exciting. In a normal small organic molecule, you will do it, signal is gone. In some of this, it will take weeks, if not months, to exchange. This tells you how hydrophobic these are. Water does not penetrate the interior. Those hydrogen bonds are so well protected that they're there for, believe me, months. We were published on this. And so we are using, maybe not recording deuterium NMR, but indirectly utilizing to get insights into dynamics.
And one more question. So the structure changes if the pH
yes.
is changed.
This is
so
one
does
of...
it mean that when I'm nervous or somebody, how to say, under stress, locally,
this is excellent
these changes occur?
This is excellent point, and we actually also think this way. How formation or one of the other will expose certain nucleotide to oxidation. You're very right. You're very right. So now, how to utilize this in living cell, like very complex phenomena? We have to think these are molecules that are in some very important biological context, but I have to admit, they're in test tube, they're in vitro environment, right? How it happens, but you're very right. Formation of the structures changes these bad things we read about, oxidation stress and all this, that actually do create damage and lead to disease.
Thank you. Can we continue with some other question? So just for the end, I mean, sometimes when we do the work on our microscopy, which is not routine, we spend months and months to prepare the sample, and then microscopy takes a little time. Now, I guess in your case must be the same, this synthesis of and isolation, this particular polymorphs, be it designed structure, must be a very difficult task, isn't it?
That's why I said that some of my students have golden hands. They're trained very well, but you need this something extra. I guess if you believe in your experiment, that's important part of it, right? You agree with me? I show you this one. So this is VK1. So Wojciech was a young PhD student, and he was bringing me the spectra. Everything here looks perfect, but there were the signals up here. So these sugar protons, you expect them here, not here. So after third, fourth time, so it's already taken a few weeks, right? So he's showing me back the same. So he started, oh, it's not beginner, it's actually something in it. And I also show you the structures. They're really new, but you're completely right. You need, I mean, we do work on these samples. They require attention, they require, I mean, the protocols are there, but it's what we say, I mean, it might sound a little bit odd. It's like every molecule is a little story by itself. It's really amazing. Once we know it, it's reproducible, and I try to make this point, how other people can reproduce and we can reproduce other people's work, but you need to understand it. And you're very right. Making a sample, that's the bigger part of our work.
Okay, so shall we conclude then? Thank you very much again.
Thank you.
And I guess, are we invited for some after party? I don't know.
Thank you very much. Well,